Hello everyone. Today I will discuss an integrated lecture on incretins. This is important so long as for the postgraduates of general medicine and pharmacology, and the objectives of the discussion are to understand the incretin physiology for better comprehension of their role in diabetes mellitus and use of newer agents in the management of type 2 diabetes, and to appreciate the emerging role of GIP and GLP-2 agonists in new drug development for inflammatory bowel diseases and obesity. Now, incretins are insulin secreting substances or peptides released from gut in response to glucose ingestion. The responsibility of the incretins is for early phase and late phase insulin secretion, and this accounts for about 50 to 70% of total postprandial insulin secretion. Thus, we can understand that incretins play a key role in glucose homeostasis. The effect of incretin is impaired in type 2 majority onset diabetes mellitus. Now, among the incretins, GLP-1 and GLP-2 are co-secreted in equimolar concentrations. GIP is another important incretin, and oxyntomodulin is also being studied. Now, natural incretins GLP-1 and GIP have very short half-life, and therefore they are not used clinically. Their half-life is short because of rapid degradation by dipeptidase 4 enzyme and renal clearance. However, several derivatives which are semi-synthetic in nature have been developed and are in clinical use today. Now, formation of incretins occur from a larger molecule of 158 amino acids called as pre-pro-glucagon, and is undergoes post-translational modifications in the tissue-specific manner. For example, in pancreatic alpha cells, prohormone convertase converts this into glucagon, which is 29 amino acid, and the remaining portion is glycantin-related pancreatic peptide or GRPP. In the ileum, in gut colon L cells, the prohormone convertase 1 and 3 converts into three molecules: oxyntomodulin, GLP-1 and GLP-2, and the remaining portion is also there. The almost similar things occur in brain, particularly feeding center and nucleus tractus solitarius, and there is a central formation of incretins. Now, GIP or glucose dependent insulinotropic polypeptide is the incretin which is active physiologically, although it is not very potent in releasing insulin. Now, this works in close concert with GLP-1. It is glucagonotropic in a glucose dependent manner, meaning thereby that under high glucose conditions, that is hyperglycemic conditions, it stimulates the release of insulin and lowers glucagon level. Conversely, under euglycemic or hypoglycemic condition, glucagon levels are increased. Therefore, it plays key role in glucose homeostasis, maintaining ideal levels of glucose. Now, fat cells have GIP receptors in abundance, and thus GIP plays role in fat deposition. GIP secretion is dependent on formation of chylomicrons, and GIP promotes fat deposition by lipogenesis and fatty acid uptake in fat cells. GIP also potentiates the central appetite suppressant effect of GLP-1. We will study this later. Oxyntomodulin is another ingredient which is increasingly being studied nowadays. Now, this is called so because it is modulates secretory function of the gastric oxyntic cells. It is also found by post-translational processing of pre-pro-glucagon. It comprises entire 29 amino acid sequence of glucagon and has additional 8 amino acid carboxy terminal sequence also. That this incretin has many actions which are similar to GLP-1 and GIP. Therefore, it can act as a dual agonist in a manner that it stimulates GLP-1 receptors and receptor for glucagon. It reduces appetite and also reduces weight. Levels of oxymodulin are reported to be low in new onset diabetes, that is secondary diabetes after pancreatitis. Development of dual agonists of these receptors, that is glucagon receptors and GLP-1 receptor, represent a new therapeutic approach for diabetes and obesity. And in near future, oxyntomodulin receptor acting drugs will be available for these conditions. Now, if we see GLP-1 and GIP and compare them, synthesis is common, that is post-translational modification of pre-pro-glucagon. The amino acid sequence of GLP-1 is 30 amino acid, whereas GIP contains 33 amino acids. GLP-1 is secreted by L cells of the ileum, colon, and rectum, whereas GIP is secreted by K cells of duodenum and proximal ileum. Therefore, secretory sites are different in gut. Glucose, fats, but not proteins are the potent stimulators of the secretion of GLP-1, whereas glucose and fats are stimulators for GIP. Now, overall effects of GLP-1 are that GLP-1 acts on the receptors which are G protein coupled receptors located in pancreas, gut and brain, and there is decreased glucagon release from alpha cells, increased insulin release from beta cells, and decreased gastric emptying. The effects of GIP are more or less similar to some extent. It also acts on G protein coupled receptors for GIP. Glucose levels determine increase or decrease in glucagon secretion by GIP in a glucose dependent manner. Increased insulin release is there and gastric acid secretion is reduced. In type 2 diabetes, insulin response is impaired both for both of these incretins. Now you can see in this pictorial that effects of GLP-1 on pancreas are marked by increase in insulin secretion, decrease in glucagon secretion, decrease in beta cell apoptosis, and increase in beta cell proliferation. So it has got beta cytotropic action in general with effect on the alpha cells. Almost similar effects are also seen with GIP, but there is increase in glucose, increase in insulin, and increase in glucagon, decrease in beta cell apoptosis, and increase in beta cell proliferation. And both decrease gastric emptying and gastric acid secretion. There are central effects. There is decrease in the food intake by GLP-1. There may be increase in memory with both. GIP may also have a key role in fat accumulation, whereas GLP-1 may have beneficial cardioprotective actions, and GIP may have some effect on the bone formation. Postulated mechanism of GLP-1 secretion is that the plasma GLP-1 levels rise upon intake of the food, probably because food induces stimulation of the sweet taste buds on tongue that initiates a neural reflex which goes to L cells. That means the enteric nervous system is directly or indirectly involved. Now, presence of glucose in the distal intestine also increases GLP-1 secretion, and glucose-induced closure of potassium ATP channels in L cell secretes GLP, and also the other one is the SGLT1. The mechanism of GLP-1 action is that it acts on the G protein coupled receptors which are present widely in body tissues, particularly on alpha and beta cells of the pancreas, the gut, and the brain cells. GLP-1 increases cyclic MP level in beta cells, and thus insulin release is promoted. And this insulin release is during first and the second phase of the food intake. There could be a direct action of gastric musculature also, and there is a relaxing effect due to indirectly release of the nitric oxide from enteric neurons. In addition, GLP-1 also stimulates sensory endings of the vagus during meals. Now, several substances act as GLP-1 secretogogues, glucose being the main secretogogue, gastrin-releasing peptide, long-chain fatty acids, leptin, some neurotransmitters and neuromodulators, GABA, somatostatin, alpha-beta agonists also release GLP-1. And in fact, GIP is also involved in secretion of GLP-1. Now, so far as long-chain fatty acids are concerned, there is increased CCK from the eye cells of the intestine, and this increased CCK binds with the receptors which stimulate GLP-1 secretion. Now, physiological effects could be that GLP-1 is actually secreted continuously with meal-associated surges, and the receptors which are found in pancreas, fat cell and brain are stimulated. Normally it is metabolized by DPP4 enzyme and other endopeptidases found in these cells, and some of the metabolites are also active. There are four characteristic effects of GLP-1 which should be understood. Number one is increased glucose dependent insulin release from the beta cells, decreased glucose secretion from alpha cells, acting as a neurotransmitter in brain to induce satiety and cause loss of appetite. Therefore, it may produce central anorexic action leading to weight loss. And of course, it delays gastric emptying. It reduces secretion of pentagastrin and acid secretion in response to food. And it is also found to have beneficial cardiovascular actions. So these actions are important as far as physiological role of GLP-1 is concerned. Now, insulinotropic action of GLP-1 acts as a regulator for blood glucose level maintenance, and it amplifies postprandial insulin synthesis and secretion. There is increased satiety by central action, which reduces food intake and fluid intake in normal and obese individuals. In fact, actually, some of the analogs have been primarily indicated to reduce weight in obesity, and their effect is significant. Inhibition of gastric emptying and acid secretion allows retention of food by a relaxation of fundus. Therefore, gastric emptying time is increased. Vagal involvement is there because denervated vagus in animals does not show this effect. Pancreatic beta cells proliferation occurs under the influence of GLP-1, and there is increase in the beta cell mass. Therefore, insulinotropic and beta-cytotropic action of GLP-1 is important in maintenance of glucose homeostasis and favoring glycogen deposit in liver. A similar proliferative, anti-apoptotic and neogenic effect have also been found on the neuronal cells. That means neuroprotective effect could be there. As I spoken earlier, several GLP-1 agonists are already being used in the clinical management of type 2 diabetes alone or in combination. Now, GLP-1 can be compared with GLP-2, which is also co-secreted in equimolar concentration. Its amino acid sequence is different. Total amino acid content is 33, and it occurs in two forms, as GLP-1 also occurs in two forms. There are separate receptors for GLP-2 also, and their chromosomal location for control is different. GLP-1 is chromosomal location is 6, that means messenger RNA is controlled through 6 band 2T1.1, whereas chromosome 17 band 13.3 controls GLP-2. Now, DPP-4 inactivation is very rapid for GLP-1, but it is slow and low level for GLP-2. Both have very short half-life, but GLP-1 half-life is much shorter because of rapid inactivation by DPP-4, and renal excretion occurs in both these conditions. Now, among the similarities and differences, despite being co-secreted, some of the effects are different. For example, GLP-1 reduces chylomicron production and reduces triglycerides, whereas GLP-2 increases postprandial chylomicronemia and also increases formation of triglycerides. Many of the organs have GLP-1 receptors, where distribution of GLP-2 receptor is mainly in the intestine, stomach, and a few centers in the brain. And the main role of GLP-1 should be understood as glucose homeostasis, whereas GLP-2 is for promotion of growth and function of the intestinal mucosa for adaptation and repair. So it has intestinal cell protective effect rather than glucose homeostatic effect. Now, physiological actions of GLP-2 have been studied. The main effects are on GIT, and because their effects are on GIT rather than glucose regulation, some do not consider it to be an incretin like GLP. So it is different than GLP-1 incretin. The main function is to maintain integrity of intestinal mucosa, and this insulinotropic effect is shown by increased intestinal growth due to enhancement of the crypt cell proliferation, inhibition of apoptosis, and therefore villus height is also increased. Therefore, intestinal repair, regeneration is promoted by GLP-2. It also enhances nutrient uptake required for cell growth, particularly fluid and electrolyte, by augmenting the transporter activity, protein activity, by increased expression of enzymes and transporters in the intestinal cells. Now, the proliferative effect is evidenced by increased mesenteric blood flow and reduced gastric motility. Neuroprotective effects have also been found, and selective uh, agonists have also been developed. One of them is teduglutide, which is DPP-4 resistant and have been developed by substituting glycine in place of alanine in the second position of the GLP-2 molecule. It is given subcutaneously and been undergoing the clinical trials for short bowel syndrome, which is a GIT disorder. Almost similar is ZP1848, which is under investigation and is being developed for the treatment of Crohn's disease or inflammatory bowel disease. Since GLP-2 has the proliferative actions, one suspects that a possible hazard could be promotion of the growth of polyps if present, or may cause malignant changes, carcinoma, colon, and it has been evidenced in the animal studies. Therefore, the key points of this discussion today are GLP-1, GLP-2, GIP, and oxymodulins are the important incretins, and among them, GLP-1, GIP, and oxyntomodulin are three important incretins. Now, the four important actions of GLP-1 are characterized now, and these include increased glucose-dependent insulin release and release from the beta cells, decreased glucose secretion from alpha cells, acting as a neurotransmitter in the brain to facilitate satiety and decrease appetite, and therefore promote weight loss. And several agonists have been developed for the treatment of type 2 diabetes, and some of them are also developed for the treatment of obesity even without diabetes. GIP and GLP-2 agonists are being evaluated for GIT disorders. Interested readers may find these key references very useful. And thank you very